Hello and welcome everyone to this podcast for epigenetics. In this podcast we're going to talk about histone modifications. As we get started, I want to talk about this hypothesis called the histone code hypothesis. If you remember back in your days in genetics class, you learned about the genetic code. And the genetic code contains 64 different possibilities of these four bases, A's, C's, G's, and T's. And in combinations of three, they would indicate which amino acid would be made. AUG would give us methionine, GGG would give us glycine, and there's a separate code for every amino acid. The histone code hypothesis is similar, and I want to write it out first, and then we'll talk about it. The histone code hypothesis. So this hypothesis is stated as such. The transcription of genetic information found in the DNA sequence is at least partly regulated by histone modifications. In essence, what we're saying is that this histone code will help explain the epigenetic code. Now let me draw it out for you in a slightly different way. Just like the genetic code has A, C's, and G's and T's, the histone code has H2A, H2B, H3, H4, and to some extent H1. Remember, H1 is that linker histone protein that's not part of the octamer. And these histones can have various modifications on them. Maybe a methylation, maybe a ubiquitination, maybe a biotinylation, maybe an acetylation. Could be a phosphate added to it. Could be a sumo modification. In the way that these histones are modified within the nucleosome will help us predict which genes get expressed. Because remember, we have our DNA here, and we have histones packaging that DNA up. And the genes hidden within these nucleosomes are protected because of the way these histone proteins surround it. Just like we learned last week that the DNA can be methylated. And as I stated up here, what we're going to talk about today is how these histone proteins can be modified. And how these various modifications shown here, up there, I should say, methylation, phosphate, phosphorylation, acetylation, and so forth. These different modifications will either hold the chromatin together tighter to prevent transcription, or it will loosen it up, allowing for transcription to occur. Okay, I'm going to go over some nomenclature for how we describe histone proteins. We've talked a lot about histones already. We've talked about H1, H2A, H2B, H3, and H4. And we talked about how these are part of the nucleosome. And up here is the linker, just to remind you. All of these histones are proteins. So it has an amino end and a carboxyl end, and all the amino acids in between. And they're numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, and then all the way down the line. So if I'm talking specifically about the 9th amino acid of histone 3, and this 9th amino acid of histone 3, let's say, is a lysine, I'm going to write it like this: H3K9. Histone 3 and lysine at position 9. This way everybody knows what I'm talking about. And then I might say this has been modified with an acetyl group, or maybe with a methyl group. However it's been modified. But we always like to say which histone we're talking about and which amino acid of that histone we're talking about. So on the previous slide we talked about the different molecules that can be attached to the histones to modify them. We're going to spend some time talking about each of those modifications, but before we do, I want to draw a piece of DNA here, like we often do. I'm going to draw a histone protein on it. I'm just going to draw it like this. And as you know, they're evenly spaced along the DNA. Like most proteins, they're fairly globular. They're folded in a very energetically favorable manner. But one thing that they all have is protruding from them is the N-terminal tail. I'm perhaps exaggerating the size of the tail here in comparison to the whole nucleosome, but I just want to make it clear that this tail is long enough and is exposed enough to be modified. So when we talk about modification of the histones, we're almost exclusively talking about modifying, adding these covalently bonded molecules to this N-terminal tail, because it's the one part of the histone that is fairly exposed. And as I said previously, these modifications may tighten this association, keeping transcription turned off, or it may relax it, releasing the DNA a little bit, that all those enzymes necessary for transcription can have access to the DNA. But it is this N-terminal tail that we will be focusing on. And the first one we're going to talk about is acetylation. And let me write that down here. As the name implies, acetylation is the act of adding an acetyl group. So this is what an acetyl group looks like to the histones. And in this case, they are very specific. They usually will add these acetyl groups to lysines. Or we might just write LYS, but more often than not, for simplicity, we'll use the single letter code K. You don't need to memorize all the single letters of the amino acids. You might for other classes, and some of you may have over the years. But while you're taking the exam, you may want to have a list printed off next to you while you're answering some of these questions. I should also mention that in some cases, acetylation can occur on arginines, which has a single letter code of R. Now, what do they do when they bind? Remember that arginines and lysines, but we're really going to focus on lysines. They're the ones that are most often acetylated. Lysines have a positive charge. These positive charges here are important because remember, DNA by and large carries a negative charge because of those phosphates. And this helps form strong electrostatic interactions between these two positive and negative elements. So if we look at our drawing, what happens is these acetyl groups here will bind to these lysines here, and they neutralize the charge. When they do that, the DNA here will loosen up. The histones will still be associated with the DNA, but in a much looser association. I'm going to draw the histones first here, and then the DNA, and I'm just looping it out here to show you how it's exposed a little bit more because of the acetylation of that histone. And I guess to make it legal here, let me show the N-terminal tails here. Acetyl groups attached, neutralizing that positive charge, allowing this DNA to loop out some. Up here you can see I drew it really close to the histones. Down here you can see I drew it exaggeratedly looped out. This would allow various enzymes now to have access to this DNA, to modify it, further modify the histones, to start to transcribe the genes. So this is just one successful strategy that can be used to relax the DNA. Now, I should mention a very important point, and that is that this is reversible. So this here is adding the acetyl groups, but it is reversible, and we can remove the acetyl groups just as easily. And I'm going to write this as a deacetylation. So it is reversible. Now while we have this written here, I want to talk about one of these histone lysines that is commonly modified, and that's histone 3, lysine 9. I bring this one up because it brings up an interesting point. This particular lysine can be acetylated, like I just said, and when it's acetylated, we would expect an increase in transcription, TXN for short. But this same lysine can be methylated, and when it does that, it decreases transcription. We'll talk more about methylation in just a little bit, but we'll learn then that methylation can sometimes increase and sometimes it can decrease transcription. It causes lots of interesting problems for us. So this site here is competed for by various modifying enzymes. Some that might add an acetyl group and some that might add a methyl group. Okay, I want to talk about this neighboring gene problem, and I'm going to draw the DNA this time like so, where we have one in here, 5 prime, and the other in 3 prime. I just drew it a straight line, not as a double helix, but you should recognize that this would of course still be a double helix. I also intentionally did not draw it in just a straight linear line because I'm trying to make a couple points here. First, let's draw some genes here. And let's say of these genes, this gene here, which we'll call gene 3, very clever of us, and we'll call these genes 1, 2, and 4. And as I said, let's say gene 3 we want to be expressed. And to make it expressed, the histones around it are going to be acetylated. So they have these N-terminal tails coming off here, and they've all been acetylated, loosening the interactions of these histones so that enzymes can come along here and turn the gene on. However, these genes here are off. In fact, but these other genes are off, and they have histones, of course, all around them as well. And they have those N-terminal tails that are not acetylated. How do we make sure that the compaction of the neighboring regions here, say here, doesn't compress this piece of the chromatin? Remember, euchromatin is very flexible. This region here could come right along here, and it might, coincidentally or accidentally, interact in such a way that it would prevent transcription. So the cell needs a way to reinforce active gene boundaries. Some way to say, almost like to build a structure here saying this is expressed, the rest of you don't bug this area here. How does the gene, or how does the chromatin do that? How does it reinforce this area of activity, making sure it remains open? So we could also say it that way, reinforces open structure of chromatin. Well, luckily there is a way, which is good since I'm talking about it. It would be a little awkward at this point if I said, no, there's no way to fix it. So what happens is there are these proteins that will come along here, and they will bind to acetylated lysines. And they'll do it with quite a high degree of specificity. They will bind to some acetylated lysines, but not others. Some of these proteins have the ability to bind to multiple acetylated lysines and still ignore others. All of this is probably to enhance the regulations of these genes. These proteins have a domain in them, and you need to know this domain. The domain is called a bromodomain. These bromodomains help reinforce this active gene boundary by saying only these genes are being turned on, and more importantly, or at least equally importantly, they bring in protein components necessary for transcription. One of these, or several of these, would be transcription factors or chromatin modifying proteins. So remember, I'm going to stress what I mean by a domain. Remember, I'm just going to draw it here. We may have a single protein here. So one protein here, but three different domains. They would probably fold up on each other, but I'm just showing it this way for simplicity. Each of these domains may have a very unique feature. One of these may be our bromodomain that binds to these acetylated pieces of protein here. Maybe the other one recruits a transcription factor, or maybe it is a transcription factor itself, binds to the DNA. Maybe the other one brings in RNA polymerase. So domains are parts of a folded protein that have unique and independent functions compared to the rest of the protein. I just want to summarize real quick what these bromodomains do. They bind to these acetylated lysines, only acetylated lysines. They reinforce this border of the gene. They're often found attached to large protein complexes like transcription factors, the SWI/SNF complexes that are important in activating gene expression. Okay, I think that makes for a good stopping point for this first podcast. In this podcast, we talked about some general ideas about histone modifications and introduced some of the basic concepts and really dived into histone acetylation. In the next podcast, we're going to talk about histone methylation and some other modifications that occur on the histone proteins. If you have any questions at all, please let me know. If not, I'll see you in class. Bye.